everyone. We are back with a very important topic for today. So, as you know, that the Bio ATP exam is due, and today we have kept this session to plan an investigation to determine the optimum temperature for the germination of tomato seeds. So, germination is the process by which the seedlings emerge from the seed. So, here we have to plan an investigation to select the optimum temperature required for germination. So how do we go about it? You very well know that you start in a sequential manner by writing the aim. The aim is to find the temperature, to find optimum temperature. Optimum temperature for germination of seeds. Okay. After you have written this, and mind you, your words should not exceed the lines. Try to fit in over there. And the materials or the apparatus required. So we will need the tomato seeds, of course. The tomato seeds. And they should be of uniform variety, of course. Petri dishes where you keep the seeds. The Petri dish. Then we would require filter paper as a base for the seeds, and to add water, distilled water, of course, distilled water to prevent from contamination. You will require incubators that have set temperature. Well, you can also use water baths, but incubators are better because they give you the exact temperature, and you can also keep thermometers. All right, so aim is clear, materials required are clear, and how do you go about the process? What do you do to determine what temperature is optimal for germination? So that means you will need to calculate the germination percentage, and wherever the germination percentage is more, at whichever temperature, that temperature would be the optimum. So how do we go about? What do we do for this? So first of all, place equal amount of seeds with equal spacing, okay? Equally spaced. Okay, equal spaced also in the Petri dishes. In the Petri dish. Okay, after placing the seeds on the Petri dish, then you would just, keep all the other factors constant. So other conditions, keep light, moisture. So filter paper, some water droplets could be sprinkled on the filter paper to keep the moisture. And remember, it should be almost constant. So light, moisture should be constant. Only the temperature varies. We would vary the temperature because we have to calculate the optimum temperature. Now, incubate the seeds. Incubate the seeds at different temperatures. You could take a range of temperatures, at least 5 readings you could take. Say, you start with a 10 degree, then you go for 20, 25, which is the optimum normal room temperature, and then 30, 35 till 40 you can take, 35 degrees and 40 degrees. There you go. Yes, so we have placed the seeds on the moist filter paper, and with fixed conditions of light and moisture, we kept it in the incubator at fixed temperatures. Sorry, with different temperatures. Now, leave them for a week. Leave them for a week. And notice or count the number of seeds germinated. Count number of seeds germinated for every Petri dish kept at different temperatures. Now, what do you calculate? You will calculate after you've got the result. You calculate the germination percentage. Okay, germination percentage rate. So how do you calculate the germination percentage rate? Number of seeds germinated in a particular period of time. So the time always is, say, a period of 7 days. So we have taken a time of 7 days, and then you calculate the germination percentage. So percentage, or you can just calculate number of seeds germinated by total seeds into 100. All right. So at every temperature, you need to calculate this germination percentage. Now, you can plot a graph. You can plot a graph of the germination percentage versus temperature. Identify the peak. Identify the peak here. So whatever the peak comes, that is your optimum temperature. So you identify the peak. Identify the peak, and from the peak, you will get the temperature, the maximum germination takes place. So what do we conclude? The conclusion is you will get the highest peak, and that is your optimum temperature. So highest germination, identify the peak with highest germination percentage. So whichever shows the highest germination percentage, that corresponding temperature is the optimum temperature. You could also note down some precautions as to handle the incubator with care. And be careful or use same kind of seeds. All right. So the variable things were the different temperatures, and the things to be kept constant was the same kind of seed, same kind of spacing, the same time. So with this, we complete this investigation. I hope you've understood it well, and it should fit in the entire space provided for the question. Thank you so much. Stay tuned for further answers.